Welcome. Today, we're diving into the fascinating world of stem cells and aging, with a special focus on a key molecular player, NRF2. As research has advanced, scientists have discovered that redox and metabolic mechanisms are central to stem cell survival and their remarkable regenerative abilities. NRF2, a transcription factor, stands out as a master regulator of cellular redox and metabolic homeostasis. But what exactly does this mean for stem cells as we age? In this video, we'll explore the emerging science behind NRF2's role in stem cell biology, especially in neural stem cells, and discuss how this impacts aging and age-related diseases. Redox and metabolic states are now recognized as crucial regulators of stem cell homeostasis. Both pluripotent and adult stem cells have unique redox and metabolic programs that determine their survival, self-renewal, and differentiation. These programs are essential for maintaining the delicate balance that allows stem cells to regenerate tissues throughout life. Interestingly, genes involved in redox and metabolism are among the most highly expressed in stem cells, highlighting their importance. As we'll see, disruptions in these processes can have profound effects on aging and disease. NRF2, or nuclear factor, erythroid-derived 2, like 2, is a transcription factor that binds to antioxidant response elements, AREs, in the DNA. This enables NRF2 to control the expression of genes involved in redox regulation, proteostasis, DNA repair, and metabolism. NRF2's role extends beyond stress response. It influences cell proliferation, differentiation, mitochondrial function, and protein quality control. Importantly, NRF2 activity declines with age, which may contribute to age-related diseases and reduced regenerative capacity. In the adult brain, neural stem cells, NSCs, are primarily found in two regions: the subventricular zone, SVZ, near the lateral ventricles, and the subgranular zone, SGZ, of the hippocampal dentate gyrus. These niches are highly organized microenvironments where NSCs reside in a mostly quiescent state, ready to activate and proliferate in response to specific signals. The ability of NSCs to self-renew and differentiate into neurons, astrocytes, or oligodendrocytes is crucial for maintaining brain plasticity and function throughout life. Redox mechanisms are vital for the survival and function of stem cells. In neural stem cells, redox-sensitive signaling networks control self-renewal, proliferation, and differentiation. Even small changes in redox state can dramatically alter stem cell behavior. For example, a more reduced state promotes proliferation, while a more oxidized state can trigger differentiation or apoptosis. NSCs have high antioxidant capacity, allowing them to adapt quickly to oxidative stress and maintain their regenerative potential. Metabolism is another key regulator of neural stem cell function. Quiescent NSCs primarily use glycolysis and fatty acid oxidation to meet their low energy needs. When activated, they shift to oxidative phosphorylation and increased mitochondrial activity to support differentiation. This metabolic switch is accompanied by changes in mitochondrial morphology and increased production of reactive oxygen species, ROS, which further influence stem cell fate decisions. Maintaining an appropriate redox and metabolic balance is essential for NSC function. Too much ROS can be harmful, but a certain level is necessary for normal proliferation and differentiation. NSCs are uniquely equipped to sense and respond to changes in their environment, adjusting their antioxidant defenses and metabolic pathways as needed. This flexibility allows them to maintain their regenerative capacity even under stress or injury. NRF2's influence extends to many types of stem cells, not just neural stem cells. It regulates genes involved in redox balance, metabolism, and protein quality control. In human embryonic stem cells and induced pluripotent stem cells, NRF2 activity is essential for self-renewal and differentiation. Similarly, adult stem cells like mesenchymal and hematopoietic stem cells rely on NRF2 to maintain their regenerative functions. Other adult stem cell types, like hematopoietic and mesenchymal stem cells, also depend on redox and metabolic changes for self-renewal and differentiation. For instance, hematopoietic stem cells in the bone marrow rely on glycolysis and fatty acid oxidation to maintain quiescence. When these metabolic pathways are disrupted, stem cell function declines, highlighting the universal importance of redox and metabolic regulation across different stem cell populations. During stress or injury, NRF2 becomes even more important for stem cell survival and function. Studies show that activating NRF2 can enhance neural stem cell proliferation and differentiation after brain injury, while loss of NRF2 impairs these processes. This suggests that boosting NRF2 activity could be a promising strategy to support tissue regeneration and recovery after damage. NRF2 is not just about stress response, it's also linked to longevity. In various animal models, activating NRF2 or its homologs can extend lifespan, while loss of NRF2 shortens it. However, there's a delicate balance. Too much NRF2 activation can be harmful. In long-lived species like naked mole rats and certain birds, high NRF2 activity is associated with enhanced stress resistance and longevity, suggesting evolutionary conservation of this pathway. Ironically, as organisms age, NRF2 activity tends to decline just when it might be needed most. This reduction is seen in both animal models and humans, and is associated with decreased expression of antioxidant genes, lower glutathione levels, and increased oxidative damage. The loss of NRF2 function may contribute to the breakdown of cellular homeostasis and the onset of age-related diseases. Aging-related decline in stem cell function is closely tied to NRF2 loss. In neural stem cells, reduced NRF2 during middle age leads to decreased proliferation and neurogenesis, while increasing the tendency to form astrocytes. Experimental studies show that restoring NRF2 in old stem cells can rejuvenate their function, while knocking it down in young cells accelerates aging characteristics. This highlights NRF2 as a critical mediator of stem cell aging. Premature aging diseases, such as Hutchinson-Gilford progeria syndrome, HGPS, also involve NRF2 dysfunction. In HGPS, NRF2 is trapped by abnormal proteins, leading to chronic oxidative stress and accelerated aging. Restoring NRF2 activity in stem cells from HGPS patients improves their viability, suggesting that NRF2 activation could help counteract premature stem cell exhaustion and related pathologies. Cellular senescence, a hallmark of aging, is also influenced by NRF2. In various tissues, impaired NRF2 signaling accelerates the onset of senescence, while activating NRF2 can prevent it. This is especially important in stem cells, where maintaining a balance between quiescence and senescence is vital for long-term tissue health and regeneration. The decline of NRF2 and stem cell function with age is linked to a range of age-related diseases, including neurodegenerative disorders like Alzheimer's and Parkinson's. Studies show that activating NRF2 can restore neural stem cell function and protect against toxic insults in disease models. This suggests that NRF2-targeted therapies could help preserve stem cell pools and improve outcomes in age-related neurological diseases. Proper protein quality control, or proteostasis, is essential for stem cell survival. NRF2 regulates genes involved in autophagy and proteasomal degradation, both of which decline with age. Loss of these processes impairs stem cell activation and self-renewal, contributing to age-related tissue dysfunction. By maintaining proteostasis, NRF2 helps stem cells respond to stress and preserve their regenerative capacity. NRF2 controls the expression of many antioxidant enzymes, such as peroxyredoxins and glutathione-related proteins. These enzymes are crucial for maintaining redox balance in stem cells. High antioxidant capacity supports stem cell self-renewal and protects against oxidative damage, while loss of these defenses leads to impaired differentiation and increased vulnerability to stress. Beyond redox regulation, NRF2 also influences stem cell metabolism. It controls genes involved in carbohydrate, lipid, and mitochondrial metabolism. For example, NRF2 can shift glucose metabolism towards the pentose phosphate pathway, support mitochondrial biogenesis, and regulate fatty acid oxidation. These metabolic adaptations are essential for stem cell activation, differentiation, and survival. NRF2 also interacts with epigenetic regulators and transcription factors to control stem cell fate. For instance, NRF2 influences the activity of sirtuins, OCT4, and NANOG, which are key for maintaining pluripotency and initiating differentiation. Additionally, NRF2's relationship with the NF-kappa B pathway helps fine-tune the balance between self-renewal and differentiation, especially in neural stem cells. In summary, NRF2 is emerging as a central regulator of stem cell biology, aging, and regeneration. Its ability to control redox balance, metabolism, proteostasis, and gene expression makes it a promising target for therapies aimed at rejuvenating tissues and combating age-related diseases. As research continues, understanding how to modulate NRF2 in specific stem cell types and contexts could open new avenues for extending health span and improving quality of life as we age.